Right. The black moth, the peppered moth, white moth, right.
So the black one came from the white one over generations, is that what you're saying?
That's the idea, is that the evolution of the dark morph of that moth in industrial England came from a mutant moth that had a mutation that caused it to be darker than its originally white ancestors.
So were those two butterflies pictured next to each other, or were they cropped next to each other? Because I couldn't tell if there were still white ones existing with the black
So
ones.
in that picture, yeah, where did the white moth come from?
Yeah.
Do they still exist?
Yeah.
Does anybody know? I don't actually know the answer. It's a great question. So were the white moths, did the white morph, the white form go extinct? Or did they just become much rarer? Certainly at least much more rare. So whether or not that went all the way extinction or not is the question, and I'm not sure about the answer. But that's a fabulous perspective on evolution. So if there's a huge pressure for being a particular color, because you're heavily predated on if you're the wrong color, for example, then yeah, you would expect it to be kind of hard to find one of those moths to take a picture of in the first place. Other questions?
Could migration be possible instead of competition?
Migration. So what? Maybe they found one of those white moths in a different environment where there was no industrialization, and so maybe that white moth came from, I don't know, I don't want to cast aspersions on any other countries. I was going to say, what if Finland didn't have the industrialization, so there wasn't a lot of soot on the trees? I don't know. But yeah, that's another great point. So it could be that the white moths just relocated. Right? They were more fit somewhere else in the world because they didn't have that selection pressure. So what else did we talk about last class?
Adaptation and exaptation.
Adaptation versus exaptation. We talked about a few examples of both. So adaptation versus exaptation. So what's an example of an exaptation? Let's try to come up with a new one. It gets harder every time I ask the question, but I'm sure you're up to the challenge. Exaptation. What's a trait that seems like it has a use, but you're not sure if what it's used for currently is why it might originally have existed? Think of some crazy animal that you know about, or plant.
What about bunnies with long ears and longer ears or shorter ears?
Okay, so yeah, what about ear size in bunnies, sure, or any animal, for example? What's an extreme ear size?
Elephants.
Yeah, bunnies, elephants. Why are elephants' ears so big? So does it have to do with releasing heat, or does it have to do with them collecting sound and funneling it into their ears so they can hear predators from a long way away?
Maybe attracting.
Maybe they're used for mate attraction. That, see? It's not about the size of your ears, it's how you use them. So yeah, all of these are potential examples of exaptation. So we observe these traits, we think, well, that could be useful for signaling to mates your quality, or it could be useful for collecting sound, or they could be used for dispersing heat from the body. Like my example last class, the tails, I've seen elephants swat things with their ears too, so maybe they're huge fly swatters. Maybe that's why they originally evolved. Okay, great example of an exaptation. We also talked about the fact that, well, the idea that there are some vestigial traits that we have that don't disappear, appendices, male nipples, etc., presumably because there's no reason not to have them. They're not costly to us, so there's not a lot of reason for natural selection to act against those traits. So last class was all about natural evolution in natural populations. And And that's what we've read so far in the textbook. Today, it's more about, in the laboratory, how do we observe evolution and what can we learn? So I'm going to use two different examples. One is one you read about, Richard Lenski's long-term evolution experiment using E. coli. So I'm not going to belabor this point too much because you've read about it. There's a short video I'll show. And then talk about the evolution of hermaphroditism, which seems like a pretty cool trait to have. It's certainly a valuable trait if you can evolve it. The problem is maybe it's difficult to evolve the ability to make both sperm and eggs in the same organism. So if we're going to have natural selection evolve, or occur rather, in the laboratory or in nature, what are the components? What do we need to have? What are the aspects of natural selection that cause evolution? So we need some sort of environmental pressure. That is, there has to be a cost. What else?
Time, or species that we can observe over,
So time, generations.
yeah.
The trait of interest?
So we need to have a trait. We need to have variation in the trait. So hopefully this starts sounding a little bit more familiar. So if we have variation in a trait, what else do we need?
We need inheritance.
We need inheritance. So now hopefully this is starting to ring a bell. We've got variation. So we've got differences between individuals. Those differences have to be inherited. So we need heritability or inheritance of the traits. And then what's the third component? It's kind of up here already. There has to be some sort of a cost. What's the cost in terms of when we're talking about natural selection? Different variants have different fitnesses. So we called that, what did we call that? Differential reproductive success. So in nature, this is what happens when natural selection acts. There are differences between individuals due to genetic differences between those individuals. Those mutations, those differences are passed on to offspring, and then natural selection acts on those differences by causing individuals with the less fit mutations having fewer offspring over time, generation to generation. So we can do exactly the same thing in the laboratory. We can do it on a faster time scale. We can actually observe this process happening, but we just have to make sure that in our laboratory experiments, we check all three of those boxes, that there be a cost, differential reproductive success, that there be variation, and that those variants be genetic. So how would you know if a variation that you observe is genetic and not environmental?
Would it be different than the parent?
Not necessarily. So would it be different in the parent? Not necessarily. What did we talk about in class that's a way to measure whether or not a trait is heritable or not?
Normal reaction?
A normal reaction plot. Yeah, so if you want to know if a trait is heritable, you need to find out if you change the environment, does the trait change? So the first example we have is called Lenski's long-term evolution experiment. Whenever you read about this, you'll usually see it abbreviated LTEE. He's the grandfather, the godfather maybe? I don't know, of the idea of doing long-term experimental evolution in the lab. What this means is you take an organism, well, I don't know, what does the long-term part mean? You take an organism that has a really short generation time, really short, and you let it grow for thousands, tens of thousands of generations in the lab, and then you watch what happens. Why do this with bacteria and not elephants? Short generation time. Doubling time of like half an hour, an hour and a half. Yeast works well for experimental evolution. Something you can grow in the laboratory that reproduces really rapidly, so you get lots of mutation happening. Every generation, new mutations arise, they get passed on to offspring. So really quickly, in these tiny organisms where you can have millions and billions of them in your laboratory at a time, evolution moves really quickly, as opposed to elephants, which have a couple offspring. Gestation time is really long. We'd have to have one scientist live for, I don't know, thousands, tens of thousands, hundreds of thousands of years to observe the same amount of evolution in a long reproducing organism as we can see in a matter of years in bacteria. So that's why he focused on bacteria. By the way, this is Rich Lenski there in the middle of the photo. So he became famous for doing this experiment, which is, he started a decade or more ago and is still going on today. The only problem is the longer he runs this experiment, again, which you read about, so you'll see a few details in the movie, but the longer he runs this experiment, the more freezers he has to have to freeze samples of his bacteria. So he needs to keep getting funding from the US government to buy more freezers and have more lab space and more power to make sure that all of these lines of bacteria that he stored over the years are still available for scientists to use. There's a cost to doing this experiment, but it's been really powerful. So I'm just going to show you a quick video, 5 minutes or so, an interview with Rich about evolution. And we're going to come back to the key, so focus on what you think the key message of this video is. We'll talk about this by the end of class and more next class.
Richard doesn't have billions of years to watch evolution in action, but he does have the span of one lifetime, which he has dedicated to performing the longest evolutionary experiment in the history of science.
Into the lab every day, and from all of these flasks, they take a little bit of E. coli from one flask, they move it to the next generation of flasks. And every 1,000 generations or so, something like that, they take a sample of the E. coli, they store it in their freezer, so they can always go back in time, essentially, by looking at E. coli that lived 20 years ago, still frozen in their freezer. They can revive those E. coli, and then they can compare E. coli 20 years ago versus its descendants 20 years in the future. And they can compare their fitness. They can ask which one grows faster. They can sequence their DNA and say, okay, what mutations have occurred in the last 20 years in this one lineage of bacteria? But what's the point of this video? So,
one mutation is kind of based on the last, like, successive mutation?
So sometimes the thing that, if evolution had foresight, which it does not, if evolution was a process that could actually see into the future and know what it was trying to do, trying to accomplish, you would describe evolution as a process, sequence of mutations, all of which together give you a new trait. So in this case, does anybody remember what the sugar was that the E. coli started consuming? It was originally glucose. They fed all of the flasks growing all of the E. coli had glucose, which is a typical carbon source for E. coli. But there was a second carbon source in all of those flasks the whole time the experiment was running, from day one. I think it was citrate. And after decades, as this video explained, one of those flasks of E. coli evolved the ability to consume whatever this alternative carbon source was, citrate or something else, in addition to the glucose. The glucose was always there, the E. coli could always consume glucose. But it took that long, it took that many generations of growth for all of the right mutations to occur in that one lineage of bacteria to allow them to turn on the enzymes that were necessary to metabolize to eat the new carbons, the carbon source that had always been there. The problem was the bacteria hadn't had the enzymes to deal with that carbon source that they could access for additional food. So when they came into the lab early that day and they saw that one flask was darker, thicker with bacteria than the others, that's what had happened. That last mutation that finally let that bacterial strain eat that new carbon source had happened. So put in one sentence, what's the take-home message? Is it simple to evolve traits? Not sometimes it is. Most of the time, any trait that would be beneficial would require multiple mutations to happen to make the necessary cellular changes for whatever trait would be beneficial. So it takes a lot of time and a lot of generations to evolve, often. So these were plots from your textbook, data taken from published literature. So looking at, for example, relative fitness. And we haven't, again, talked yet about how to measure relative fitness. If you want to learn about how Lenski did it in his studies, you can go read his papers. But essentially the idea is relative fitness. What does this mean? What does it mean to have fitness in the first place? You reproduce. So I've got 2 kids, 2 daughters. I'm fit, right? I've reproduced, I've passed my genes on to the next generation. Does anybody in here have more than 2 kids? No? That wants to claim it. Okay. So in relative fitness in this room, who's top dog? Me.
But I have 2 kids.
So yeah, we're in competition. So we each then have a relative fitness that's the same. Compared to, right, relative means compared to something. What did they compare to get relative fitness? They're looking at an E. coli from 8,000 generations here. They're measuring how many offspring, for example, in 24 hours. Let's just say, this is not what they did. We'll say that the measure of fitness is how many times a bacteria divides in 24 hours. That's something of a measure of how many offspring it produces. Relative to what? Take an E. coli 8,000 generations in, you say, okay, you made 8 cell divisions in 24 hours. You're fit, but how fit? Relative to who? So is it other bacteria at the same time point? So that's what we're seeing here. Each of these different lines is a different strain. So like strain A, strain B. So at any one time point, there's a lot of variation in relative fitness. Right? Some of the independently parallel evolving bacteria are more fit than others at any point in time. They all started out at the same relative fitness, 1. So all the bacteria were equally fit because they were genetically identical. That was the first day the experiment started. And since then, all of these different parallel cultures of bacteria have been evolving separately. So there are different fitnesses. But when you calculate relative fitness, you compare a worm, say, at 8,000 generations with the ancestor. So you say you go back in the freezer, you take out the time 0 cryovial, the tube that's holding those 0 generation individuals, and you see how many offspring can they produce in a 24-hour period. And at the exact same time, on the exact same plates, the same day in your laboratory, you run the parallel experiment. With the current strain, how fit are they? So what's happened to fitness of all of these strains over 8,000 generations? How have they changed relative to their ancestor?
They're more fit.
They've all become more fit, positive slopes. At the same time, they also show up here that cell volume has increased in all the strains over time. You might use cell volume as a measure of fitness as well. So there are different ways to measure fitness. But in all these cases, it's clear that all the lines are different, all of the different cultures of bacteria that have been growing independently, but they're all increasing in fitness. This is one of Rich Lenski's students with some of the many, as you saw in the movie, petri dishes that were also used in completing this and continuing this research project. So you can do fun things in the laboratory, it's not all work. Does this experiment have all of the 3 requirements for natural selection to occur? Is there variation between bacteria? And if so, how? Mutation. So we had parallel, different cultures, different flasks of bacteria, all growing independently. So different mutations were happening as DNA was replicated and copied and mistakes were made over time. Because they're mutations, that means they're inherited. So mother bacteria passes it on to daughter cells. And there was differential fitness. So they saw that different strains were more fit or less fit relative to their ancestor over time. So when anybody asks or challenges the concept of evolution, that there's an identity by descent, that there's inheritance of changes that cause adaptation, most people now point to examples of experimental evolution, like, for example, Rich Lenski's experiment, because you can actually observe these changes, and since then, since this movie was produced, and currently ongoing, they've actually gone back and sequenced the DNA of these bacterial strains to actually pinpoint, as the movie suggested, they can identify which mutations have occurred. And they know how those mutations work and how those mutations led to the ability for these bacteria to access this new carbon source and to become more fit. And that's why experimental evolution is so cool, because we can use it to actually show evolution happening in real time. It's not a guessing game of what was the ancestor of this group of species, what did it look like, for example. We can't hop in our time machine and go back in time and observe that. But with these cases, especially when you can freeze down the bacteria and revive them thousands of generations later, you can actually, in the lab, quasi go back in time. Any questions about Lenski's experiments? The main concept here again is that multiple mutations are usually necessary to cause adaptation, to enable adaptation. So sometimes that process takes a lot of time. So here are my favorite worms again, the Caenorhabditis nematodes. They're cool because they're hermaphrodites. So here we have a hermaphrodite picture of a hermaphrodite worm. These big cells are all oocytes, so embryos. This is a hermaphroditic individual. There are 2 sexes, hermaphrodites and males, in this species, C. elegans. So that worm happens to be C. elegans. And if you zoom in using the microscope, you can't see here, but you would see sperm. And that's self-sperm. So this one individual has a gonad that can produce both oocytes and spermatocytes in the same body at different points in time. So hermaphrodites start out life producing sperm, and then at some point during development they stop producing sperm and they start producing oocytes. And when they start producing oocytes, their sperm that they already made fertilize those oocytes, and they self-reproduce. We're going to talk in a week or two starting to talk about phylogenetic trees and how to analyze them, relationships between species. So that's what's shown down here on the bottom. So the relationships over time, with time moving down this axis to present. It's kind of like a family tree of Caenorhabditis nematode species. So what really piqued the interest of evolutionary biologists when they started studying these different species same genus, all Caenorhabditis, just different species. They discovered that 2 of the species, both C. elegans and C. briggsii, have the 2 sexes, one sex is male, one sex is hermaphrodite. All of the rest of the species, what are the sexes? They're male-female. So Romanii has females, not hermaphrodites. Brenneri has females, Japonica has females. So what we'll talk about when we get to phylogenetics in this class is how to look at this distribution of traits, the 2 species that have hermaphrodites instead of females, 3 species that have females instead of hermaphrodites. We're going to try to figure out how we can reconstruct what happened during evolution to cause those traits to evolve. And it turns out in this case, I'm going to draw blue slashes across branches of the tree where this trait evolved. So it suggests the distribution of these traits, where the females exist in this relationship of all the species and where the hermaphrodites are, suggests that hermaphroditism evolved from a common ancestor that was a hermaphrodite, or a female, sorry. So way back in time, if we went back in time in our time machine, we looked, whatever worm was existing back here would have been a female. And as time passed and species split, were created, there was a point here in an ancestor of current day C. briggsii that all of a sudden gained the ability to start making sperm in a female body. Female became hermaphrodite. And that separately, at some point in time, totally separately, on the branch, the descendants of, or the ancestors rather, of C. elegans, the exact same thing happened. At one point in time, different species, one day there was a mutation or a series of mutations that allowed those ancestors to turn from females into hermaphrodites. This is really cool because it lets us test hypotheses about how evolution works. The main hypothesis tested here, and the reason that people study this group of organisms and how hermaphroditism evolved, is because when nature does the same thing multiple times, like allow hermaphroditism to evolve, one of the questions we get to ask is, if we really study this carefully, did evolution happen in exactly the same way in C. elegans and C. briggsii? Right, the critical point being, and the specific point being, did the same mutations occur to allow hermaphroditism in C. elegans that also allowed hermaphroditism to evolve in C. briggsii? Did evolution repeat itself? Or were there separate ways to evolve the same trait? So are the mutations that allow hermaphroditism in C. briggsii totally different mutations to different genes than the solutions C. briggsii found to the problem of evolving hermaphroditism? How many mutations do you think it takes to become a hermaphrodite if you're a female? 1? More than 1? I think. Who says you can become a hermaphrodite if you're a C. elegans or a Caenorhabditis ancestor? 1 mutation can make you a hermaphrodite. Got 1 vote? Multiple mutations. It's going to be pretty tricky to evolve hermaphroditism. Well, that's a good place to start, but then when scientists looked at this, they said, well, 2 out of these 5 species are hermaphrodites. So what does that suggest about how easy it is to become a hermaphrodite? I mean, I haven't told you how much time this takes, but the evolutionary biologists that started working on these species were thinking, maybe the ancestor is predisposed. That is, like in Lenski's experiments.
So they already have the genetic material to become hermaphrodites, and only
Or they're almost there.
Oh.
So Lenski's experiments suggest there are multiple mutations usually necessary to reach one evolutionary goal, a trait that's adaptive. So this suggested to scientists that, well, yeah, maybe the ancestor here had a bunch of those mutations already. And from that point forward, then, these different lineages were able to pretty easily, pretty quickly, evolve hermaphroditism because maybe it took from that point only those last 1 or 2 changes necessary to become functionally hermaphroditic. Sorry, I hope nobody's hungover because when the projector vibrates like that, it's a little bit unfortunate. For a female to become self-fertile, it's not just necessary for her to be able to produce sperm, but she also has to find a way to turn on those sperm, to activate them. So worm sperm are not flagellated. They crawl with pseudopods, so this is what sperm look like once they've been activated. They sort of crawl around looking for a oocyte to fertilize. So at least two transitions that have to be made to turn from a female into a hermaphrodite. To be able to make sperm, you have to be able to activate them so that they can actually fertilize an oocyte. Yeah?
How did you say the sperm was activated?
It's chemically. So in this case, the exposure of the sperm to a particular chemical that males have in seminal fluid activates them at ejaculation for male-female species. And in the hermaphroditic species, what I'm saying is then females have to evolve some way to expose their self-sperm to some sort of chemical that would also turn their own sperm on. So that leads me to have to conclude with a sad story. The greatest experimental evolution, in my opinion, the greatest experimental evolution project experiment that never worked. So by the end of class, I just want you to be able to tell me, why did this not work? So I've given you the background. I've talked about C. elegans for a while now. So the project was to evolve a hermaphrodite from a female. So it's simple. The hypothesis is that you can turn a female into a hermaphrodite with some mutations. Be that one mutation, which I'll indicate as an asterisk, or maybe a couple or a few. That's the goal. You start with one of those male-female species. C. ramanii, for example, doesn't matter. And magically, somehow, turn them into hermaphrodites and watch it happen in the laboratory, just like Lenski did with bacteria. So you've seen male-female mating. It was male hermaphrodite, but it doesn't matter. Males and females mate the same way. So what does it take for sexual congress to occur in worms? So the male has to be able to physically, males don't have eyes, sorry. They have to actually feel with their tail, that's where their sensory organs are, in their tail. They have to physically feel for and find a female and internally fertilize. So they have to actually be in physical contact, males and females do. Not shocking, right? Okay. So here was the concept of this experiment. We need variation. It needs to be heritable, and there needs to be differential reproductive success to evolve a hermaphrodite, right? The 3 main characteristics. So how are we going to get variation? Genetic variation. So you need to have, well, so how do you get mutations, essentially, is the question. So we need genetic variation. We start with one population of worms on a petri dish. They're all practically genetically identical. So the first step is mutagenesis. So you throw a chemical that causes DNA mutations on a population of worms. You generate all sorts of random mutations. That's how you get your starting variation. In this case, I think this actually happened, right? It didn't work, but this experiment actually took place. I think it was a chemical called EMS, ethyl methane sulfonate. It causes DNA mutations. So you take a huge population of females, you throw this chemical on them, all sorts of random mutations happen in their DNA that gets passed on, those mutations get passed on to their kids and so forth. So there's variation, it's heritable variation, it's mutations. So then the question was, if you have, let's say you start with a population of 1 million worms. How would you find the needle in the haystack? If you were successful and you made enough, the right combination of mutations in one female to turn her into a hermaphrodite, how would you find that one hermaphrodite among 999,999 females? It's always the problem with genetics experiments and experimental evolution. How are you going to find, when it does work, how are you going to know? This was the really elegant part. If it had worked, it would be elegant. You put these worms in liquid, in a flask. You mutagenize the hermaphrodites, females. You throw them in this flask where they can grow and reproduce. Wait, no, they can't reproduce because if worms are growing in liquid, what can't happen? Right, so how are males going to find a female or hermaphrodite and mate with her if they're all being shaken around in solution? It doesn't work. So you expect, if you're shaking a culture of nematodes, no mating to happen because you're physically agitating them. There's not enough time for the male to actually find a female and attach to her with your spicules that come out of your tail and internally inseminate. So you throw a chemical on a bunch of females. You put females and males together in this flask, and you let it sit for some amount of time. So time passes. Let's just say 2 weeks, for the sake of argument. What happens after 2 weeks? You go in there and you look. These worms normally have a lifespan of about a week, say. So if your experiment doesn't work, what happens when you go back to the flask? Everybody's dead. There was no mating, so there's no children. But what happened if that 1 in a million worms became a hermaphrodite when you mutated her? She's going to be self-reproducing. So there's going to be a flask that's been shaking for 3 weeks. If there's a hermaphrodite in there, no physical contact needed for mating. She's internally self-fertilizing. So then you come and there's a flask full of worms. Descendants of a new, recently evolved hermaphrodite. Why didn't this work? So my postdoctoral advisor tried this before I joined his lab, and it didn't work. We took C. romanii, threw some mutagenesis at it, grew a bunch of females in culture, and after time passed, none of them had turned into hermaphrodites. So what are some reasons why? There are many possible answers.
Could shaking the bottle have killed the worms over time?
Okay, so you need to control for, did physical agitation actually do something bad to the worms separate from what we were trying to do? So did it actually damage or kill the worms? We can do control experiments to control for that. So in this case, it didn't. But it's a very good point. So before he started this experiment, he had to prove that shaking the worms didn't kill them.
You can't discretely mutate something to try to get it into something else.
Well, so the mutations that occur are random. So what's another concern?
You'll never achieve the mutation required for hermaphroditism.
So what's the probability that you get the right mutation? So maybe we didn't use enough worms yet. Or maybe we didn't put enough chemical mutagen on them yet to hit that right mutation. Or beyond that, instead of just one mutation, go ahead.
Is it like not entirely isolating it, that it's around males, that it still has the switched on pretty much as to being females, so you're not getting...
So is there a maybe there's a change left to occur?
The environment setting wasn't.
Okay, so maybe the environment wasn't somehow tuned properly for this experiment to work, certainly possible. Eventually, my advisor came to the conclusion that there just weren't enough worms to hit on that right, as Lenski said, the right hand. These worms weren't dealt the right poker hand. Maybe some of the mutations necessary to achieve those two goals, making sperm in a female body and activating those sperm, maybe some of those mutations that were necessary to do that happened, but not all of the mutations. Who knows how many mutations it takes to make a female turn into a hermaphrodite? So maybe he needed to do this just more. Do this experiment again and again and again and again until you hit the jackpot, that right combination of mutations that causes the effect that you're trying to study.
How many times was that experiment?
Pardon?
How many times was that experiment?
This experiment?
Yeah.
I think he only did it once, maybe a few times.
That had to be 20 years, right?
Right. In an organism that evolved, that has a shorter generation time too. So elegans, Briggsii, these worms have about a half a week to a week generation time. So it's orders of magnitude longer than elegans, or than, okay, so a couple points to close. I know we're about a half a minute out of time, or half a minute until we're done. Later, somebody actually succeeded in doing this. Not by using random chemical mutagenesis, but they studied the genes that were known to be responsible for activating sperm and for causing hermaphrodites to produce oocytes. They made 2 single mutations, just those 2 that were already known to be important. They artificially made those 2 mutations in C. ramanii, one of the female species, and turned that female into a hermaphrodite. 2 mutations. So those worms really are really close, evolutionarily, the females, to becoming hermaphrodites. It just takes 2 changes. You just have to have the right 2 changes. And that's something that apparently by chance didn't occur in that flask shaking experiment. Okay. So a couple of concluding remarks and then we're out of here. We did reach the $500 threshold, so I'll chip in the rest of the money. We'll make a rap song. So what the plan is, if you made a donation, you might not remember how much you pledged, but on Friday and next Monday, I will collect money. And I'll bring a form you can sign so we record how much you donated, etc. But by next Monday, I need to have all the cash so that I have time to then submit from my personal account all of it to his crowdfunding campaign. Lastly, for next class, so thank you all for participating. Even those of you that weren't able to donate, that's totally fine. As I said, none of this impacts anybody's grades or anything. It's just for fun. We're still going to do this chapter by chapter summarizing by creating some sort of artistic poem, lyric, whatever. For next class, read chapter 3.5, just that one section, and then watch these 2 movies. So about 15 minutes or so, 10 minutes worth of movies will prepare us for a discussion for next class. Thanks very much for sticking around for an extra few minutes, and have a good day. Doctor. Oh, doctor. We were just getting excited about evolving hermaphrodites, so. Yeah.
So I sent a Google Doc myself. I'm not a Google leader.
Yeah.
So I just sent it. So is that good?
I need to have whichever Google, so it doesn't matter who sends it as long as it's the version of the document that everybody was working on.
Okay.
And
I sent it in Google Doc, but I think it's editable. So I don't know.
well, I'll check it out. I'm trying to keep up with all of the notifications I'm getting about all of this work that's being turned in. So.
Okay. All right. I just want to make sure it was okay if I turn it in, even though I'm not the group leader.
Yeah, that should be fine. I prefer to have it from the group leader because I think they might have a version that's more useful for me than you. But Will you send me an email and remind me you just told me that?
Yes.
And then I'll look into it. Then I'll know and you'll know.
Okay.
I'll let you know.
All right, thanks.
Yes.